let's go over enzymes as one of the biomolecules. Well, we know that an enzyme is a biological catalyst. Now we are calling them biological because these are part of the living organisms and they're actually also living, and we're calling them catalysts because they get to speed up the rate of a chemical reaction. But one important thing is that enzymes get to speed up the rate of the chemical reaction, but they are not changed as the reaction gets to proceed. So we'll say enzymes are biological catalysts which speed up the rate of a chemical reaction while they remain unchanged. So apart from them being able to speed up the rate of a chemical reaction, well, let me just say they get to speed up the rate of a chemical reaction. But how do they do that? They do this by lowering the activation energy barrier. Okay? So they get to lower the activation energy barrier. Well, what really is this when you're talking about activation energy barrier? So when you have got, let's say, this is a diagram which is showing the progress of a reaction to the energy. So the x-axis is representing the progress, and then here we have got energy on the y-axis. So you can have a reaction that starts like that, and then it has to go like that, and then get to drop. Okay, so from here, this hill that has to be taken. So this, at the beginning you have got reactants, at the end you have got products. So this energy that has to be taken in for the reaction to reach at the maximum point there. This is what we call as the activation energy. Okay, that's the activation energy. Now, for a catalyzed reaction, enzyme catalyzed reaction, the activation energy is going to be lowered. So you're going to see you're going to have something like this, and then you're going to reach out the product. Meaning that you only require a small amount of energy for this reaction to start moving. So activation energy is simply the energy that is required, or the minimum, let me say, minimum energy required to keep the reaction going or to start the reaction going. So before you supplying the reaction with this activation energy, that reaction will not be able to work, or the reaction will not be able to start because you have to overcome this hill. So what these enzymes do is that they are going to lower the activation energy barrier. When they get to do that, then the rate of the chemical reaction is going to be much faster because you would have taken just a few seconds or a little time for you to be able to reach that wall, or you would have spent only little energy for you to overcome this barrier. All right, so that is what these enzymes get to work. Now we are saying that these enzymes, they get to speed up the rate of a chemical reaction, but they are not changed at the end of the reaction. So how does that get to happen? Well, what we are going to have is, let me say this is the enzyme that we have. Let's say this is the enzyme. So we need to take note of the shape because enzymes are highly specific. So if this is the shape of the enzyme, this portion where the substrate or that thing that this enzyme has to speed up is going to attach is the active site. And the other site that is going to be called the allosteric site. The site which is not active is called the allosteric site. Okay. So the substrate that this enzyme is going to work on is going to be something that is going to look like that. So that is the substrate that this enzyme will be able to work on. So we are saying this is the enzyme, this is the substrate. So when the enzyme and the substrate now bind to each other, you are going to form an enzyme substrate complex. Well, we are not changing the enzyme. The enzyme still retains its shape the same way it is. Okay, something like that. And then the substrate has now come also and it attaches here. So this is the substrate that we're able to see. So what we have is the enzyme substrate complex. Now after these reactions get to happen, after the enzyme gets to attach to the substrate, then these are going to be broken. Okay, so what you are going to see is at the end, the enzyme will still come out just the same way it was. It does not change at all. But for the substrate, it will no longer be the same. What you are going to have is a product now. So the substrate is going to be broken. Okay, you are going to have maybe something like this. So you have seen that it has to broke. It broke at the middle there, and then you have got this. So here is the enzyme still with its own shape, and then now you have got, instead of the substrate which you add, now you have got the product at the end of the reaction. Okay, so this is what we get to have. So this is what we, why we say that enzymes are highly specific. Now enzymes are highly specific because they only take up one type of substrate, and that is what we call the lock and key model. So the lock and key model just tells us that each enzyme has got a particular shape, and that can only be fitted in by a particular substrate. So we also get to call this as a Fisher model. The lock and key is also called Fisher model. So this model was developed by Fisher in the year 1890, telling us that enzymes are highly specific. So that is what we are just from mentioning here. All right. So another model that we need to have to be able to know is the induced fit model. Okay, that's the induced fit model. Now, what does this model tell us? Well, the other name for the induced fit model is the Koshland model. Now, this model tells us that, of course, it was proposed by the same Koshland we are able to see in the year 1958. Okay, so it tells us that the shape of an enzyme is going to change slightly when it binds to its substrate. So at times the substrate might not be able to bind or fit in exactly. So what we are going to see is that the shape of the enzyme might change slightly. For example, let's say this is the enzyme. This is the shape that it has. And then the substrate is supposed to work on, let's say, is a bit like that. So we are able to see that this end here is pointed, but here it is not pointed. So when this substrate, this is the enzyme, and the substrate is going to fit in this enzyme, the enzyme is going to change a little the shape. So when it gets to do that, then the substrate that we are able to see is now going to be able to fit in. So we are going to see something like this. Okay, not necessarily how the shape was initially, but now the substrate is able to fit. And there it goes. It fits exactly. So that is what we call the induced fit theory, which was, or the induced fit model introduced by Koshland in 1958. Alright, so after the reaction is done, or after the enzyme substrate complex is formed, you are now going to have the product. The enzyme is going to change back to its original shape. So when this move out, when the substrate is broken into product, the enzyme is now going to change back to its original shape, and the product no longer fits into the active site. So for example, when you get to have this reaction happen, you might find that the product that you are going to have will look something like, let's say, like this. So it has been broken. Okay, the substrate has been broken. So there is the product. Now they can no longer fit into the active site of the enzyme. Alright. So when this happens, the enzyme is now going to be able to move out, going to get to look for other substrates for them to for the enzyme to be able to work on, because the aim is any substrate that it finds, it gets to attach to it, or the substrate attaches to it, in order to speed up the rate at which that substrate is going to be broken. Alright, so that is the induced fit theory and the lock and key theory. Now, when we are talking about enzymes, another important concept that we need to understand is the concept of cofactors. So that's the concept of cofactors. Now, really, what are cofactors? Well, cofactors are basically non-protein substances. Okay, these are not proteins because enzymes themselves are proteins. So these are non-protein molecules, and really, what's their aim? So their aim is to get to bind to the active site. Okay, I mean get to bind to the enzyme to activate it. So there are some enzymes which depend on cofactors for them to be able to work. Otherwise, if these cofactors do not bind to such an enzyme, then they are going to be inactive. Let's say here we have an enzyme. Okay, this is the enzyme that we have. So it for it to be active, it requires a cofactor. Meaning that when the cofactor is not attached to this enzyme, it is inactive. Okay, meaning this state when the cofactor has not binded to the enzyme and the enzyme is inactive, this is we are going to refer to this enzyme as an apo enzyme. That is going to be referred to as an apo enzyme. Now, when you have got this enzyme activated, how is it going to be activated? It is going to be activated by the attachment of its cofactor. Okay, so let's say the cofactor attaches somewhere there and then the enzyme is now active. So when you have got the enzyme plus the cofactor, the enzyme is going to be active and that is now going to be referred to as the holo enzyme. So what is an holo enzyme? An holo enzyme is simply an enzyme plus a cofactor. Since an enzyme alone in its inactive form without a cofactor is called an apo enzyme, therefore we can also say that an holo enzyme is an apo enzyme plus a cofactor. Okay, so that is what basically an holo enzyme is. Now, when we are talking about these cofactors, some of them they are inorganic, others are organic. Now, let's start with the inorganic cofactors. Well, when we get to say they are inorganic, meaning that they do not contain a carbon, right? So, inorganic cofactors are basically metal ions. For example, ions of iron, ions of magnesium, ions of manganese. So, these are going to be the inorganic cofactors. And then the organic cofactors, these are the ones now which are made up of organic compounds. Well, since they are organic, we expect of course for them to be organic compounds. So organic cofactors are also referred to as coenzymes. Organic cofactors, they are also referred to as coenzymes. Now we need to know that necessarily the cofactor and the enzyme, they do not form permanent covalent bonds even when they are tightly bonded. They do not form permanent covalent bonds, so they'll just be more attached, but they don't form covalent bonds like that. So meaning that anytime they can be separated. All right, so now let's understand a bit about cofactors, or these now organic cofactors which we are calling as coenzymes. So coenzymes are basically small organic molecules that can be loosely or tightly bound to an enzyme. So tightly bound coenzymes are called prosthetic groups. So when these coenzymes they get to tightly bind to enzymes, these are going to be called prosthetic groups. All right, so that is it. Now coenzymes they get to transport chemical groups such as hydrogen phosphates and alkyl halides from one enzyme to another. So that's their aim. Remember, they are what? They are cofactors, and they get to make enzymes active. For example, maybe this enzyme, for it to be active, it needs an alkyl group or a hydrogen phosphate. Okay, so meaning that for this enzyme to be active, they have to attach to these compounds. So the coenzymes, they are going to be able to transport these groups to different enzymes or between enzymes. So that's their work. Now, coenzymes are mostly vitamins. We need to take note that coenzymes are mostly vitamins. Examples we can talk about, we have got riboflavin. Apart from riboflavin, we can also talk of thiamine. We can talk about folic acid. We can also talk about other prosthetic groups such as NAD, okay, such as NADPH, and also ATP. And we also have what is called coenzyme A. So these are examples of organic cofactors or coenzymes. So we need to take note that they are mostly vitamins. Alright. So that is going to be it for this tutorial. Thank you so much for watching. Hope that was helpful. Remember to subscribe.